much, Adina. I'm just going to go right ahead into sharing my screen. Thanks everyone for sticking around after that really excellent panel that we just had for our next session, what we're calling Crossing the Chasm. And we're looking at this as an opportunity for us to really showcase what UCLA is focused on programmatically. And so what we're going to have here is 3 pitches from our Innovation Fund with 2 highlights from our new collaboration, the Biodesign Program. So here today, I'm Matthew Savary. I manage the MedTech vertical of the Innovation Fund. And here on the call with me, I have Jennifer McCaney. I'll just move from there. So here's where I wanted to highlight that UCLA has a really broad experience and a broad impact in innovation in the life sciences. It's really born out of the sheer number of institutions that we have here on campus dedicated to technological development. We have $1 billion a year in annual research awards, 400 new inventions a year, and you can see a couple of our highlighted recent successes. And even building on top of that is because we have the relationship with the UCLA health system, we have an even broader impact, both geographically and in clinical domain. And that's just really to point out that we have a lot of opportunity for that general research and development. But the highlight that I wanted to point out today with these pitches and with this conversation that I'm kicking off right here is something that most of the folks on the phone call are familiar with, which is that valley of death or crossing the chasm, where as a society, as a general community, we've gotten really good at funding general research and development that's here in this early stage over here. And we've gotten really great at this early commercialization phase. But when we're moving, especially in the medtech arena, around this proof of concept phase, because that incremental research and development tends to cost so much money, it's something that we have to build a little bit of muscle in. And that's actually where UCLA has stepped up strategically to start to fill those gaps. And I'll highlight, and I'm highlighting 2 of those opportunities today. So we have within the discovery domain, our traditional research and discovery, which is what happens at a university. But we've also developed something called the Innovation Fund that's directly focused on that proof of concept phase, where the university directly invests into really promising technologies and furthers them towards the goal of commercialization. And most recently, we have a new partner in the game of trying to help us fill up that pipeline to innovation, and that's the UCLA Biodesign Program. And I'll hand it over to Jennifer pretty shortly to let her touch on exactly what that is once I round out exactly what the Innovation Fund is. And so like I mentioned, there is this opportunity that we have for crossing this chasm between university and government funding and private sector funding, and that's where the UCLA Innovation Fund comes in. It's flexible capital up to $200,000 of non-dilutive funding, so that's going to be really interesting to folks on the phone. And we connect these teams with project and alliance managers, deep experts in the field, and we open it to all modalities and therapeutic areas. So it's open across the campus. And so the external benefits of a program like this are it actually further de-risks the opportunities that we have here on campus. And it conveys the team's ability to collaborate and hit milestones. So what we're going to have today, like I mentioned, is we have our 3 teams pitching, and this is our newest cohort of Innovation Fund awardees. And the exciting thing about them, like I mentioned, is that 2 of these teams are actually alumni / participants in the Biodesign Program. You probably have heard that term before, but I'm going to hand it over to Jennifer right now, just so she can give you a little bit more detail on exactly what that is.
Thank you, Matthew. Our mission at UCLA Biodesign is to advance leadership and innovation training for the healthcare leaders of tomorrow. Much like we just heard Terry Burke say, you begin with the end in mind. We start with the clinicians who understand the clinical problems facing our patients, our providers, and our health system today. Through this program, next slide. Through this program, we have a marquee fellowship that's divided into 2 tracks, a discovery fellowship for post postgraduate and professional trainees looking to start a new medical device program. We also have an accelerator track for faculty and staff interested in maturing concepts, much like you'll hear today. Next slide. As part of the UCLA Biodesign Fellowship, we've welcomed over 60 trainees to date. These range from undergraduates in bioengineering to MBA candidates at the Anderson School of Management to clinical faculty, residents, and fellows, as well as nurses within our UCLA health system and professionals from industry. Next slide. As part of the UCLA Biodesign Program, we call our home here at the UCLA Biodesign Hub in Southern California, which is a federally funded $1.5 million initiative here in Southern California that's supported by the U.S. Economic Development Administration. As part of this, next slide, we host hub events, much like the one you see here that we hosted last week with Maria Miller and Stephen Ralph from the WL Gore Medical Products Division. Next slide. And across the UCLA health system, our fellows, next slide, are working to advance products in pulmonary care, infectious disease across the UCLA health system in areas most impacted by COVID-19 this year. Finally, I'd like to invite you to join us in visiting the UCLA Biodesign website, biodesign.ucla.edu. Our applications open in summer 2021. And finally, last slide. Oh, that is last slide. Yeah. I'd like to start. Shall we transition?
I'm sorry?
Shall we transition to the presentations?
Yeah, let's go ahead.
Okay. I'd like to start by introducing our first cohort of presenters. Speaking for the next generation dermal filler team will be Dr. Mike DeLong, who is the Chief Plastic Surgery Resident here at UCLA Health. He's speaking on behalf of he and his team partner, Dr. Tim Deming, Professor of Biology and Chemistry here at UCLA. Mike, I'll turn it over to you.
Hello, everybody. Good afternoon. I'm Michael DeLong from the Division of Plastic Surgery, and along with my co-founder, Tim Deming from Bioengineering, we'd like to introduce you to the next generation of dermal fillers. Dermal fillers are gel products that are considered injectable implants. They are commonly used by aesthetic medical providers to augment tissue volumes. They're most typically used for facial enhancement or rejuvenation, such as augmenting the size of the lips or masking facial wrinkles. There are several products already on the market as dermal fillers, and 80% of the market is occupied by fillers made from hyaluronic acid polymers. These are names you may have heard of, like Restylane or Juvederm. However, all hyaluronic acid commercially available fillers currently have several limitations, which motivated us to develop a novel polypeptide hydrogel as a safe and improved dermal filler material with the potential to disrupt this market. Our team consists of myself and Dr. Deming. Tim is a well-published researcher and the prior chair of bioengineering at UCLA. He developed our filler product material and brings scientific and technical expertise to our team. He also has prior startup and manufacturing experience with these materials. I'm Michael DeLong, a clinician in the Division of Plastic Surgery. I completed a year fellowship at the FDA and the division that regulates dermal fillers and was selected to remain officially involved as a special government employee through that same division. I've also successfully sponsored IDE applications since my time at the FDA, and I'm very familiar with navigating the FDA regulatory submission process. I'll provide clinical and regulatory expertise for the team. We are also both current fellows in the UCLA Biodesign Accelerator track, and this project was selected as the first place winner of the MedTech Challenge last year out of 72 teams at UCLA and was recently selected as one of 3 winners for the UCLA Innovation Fund. So what's the problem and what are we trying to do? Current dermal fillers are not perfect. 3 limitations with current products are risk for vascular occlusion, which can cause tissue death or blindness, unpredictable volume from variable swelling properties, and the propensity to form delayed or chronic irritation and granulomas. Regarding vascular occlusion, dermal fillers have actually never been approved by the United States FDA for injection in the superior portion of the face, likely due to the high blood vessel density in this area that increases the risk of vascular occlusion, although this complication can occur with injections anywhere in the face. When dermal fillers are injection, it's done blindly. And if the filler material is inadvertently injected into the blood vessel, it can occlude the blood flow downstream. This occurs with hyaluronic acid fillers, which make up the majority of the United States market, because they consist of filler particles that typically exceed 300 micrometers in diameter, much larger than the size of a typical arterial passage. The picture on the screen is a 22-year-old girl who underwent filler injection around her eyes. The material entered an artery, occluded the blood flow, and caused a significant amount of skin to die in the middle of her face. For a purely cosmetic, minimally invasive procedure, this is obviously unacceptable. Another drawback with hyaluronic acid fillers is their propensity to absorb water and swell for days after injection. So why is swelling bad? One problem with swelling is that a swollen gel could cause external compression on nearby blood vessels, causing the aforementioned complications. Another problem is because providers are usually attempting to make subtle, symmetric augmentation that's harmonious with the overall facial appearance, precise volume of injection is critical. Consequently, providers have much less control over the ultimate result because they have to account for this unpredictable swelling of the product. The graph on the screen is from an animal study in which a hyaluronic acid filler was injected into a rat. And as you can see, over a period, the bottom is weeks, over 4 weeks after injection, the filler material swelled up to about twice the size of what the physician injected. Another significant problem with existing fillers is their potential to cause delayed or chronic tissue irritation or granulomas. It's not clear why this occurs, but it's thought to potentially be because of residual gram-positive proteins left in the filler material, because all these fillers are synthesized using cellular synthesis mechanisms from streptococcal species. While this may not seem like a major issue, I would reassure you that cosmetic patients who desire an improved facial aesthetic would notice a persistent blemish or lump in the middle of their face. These problems with existing fillers are all potentially solved by our synthetic polypeptide hydrogels. Let me first give you an overview of our product's design. Our fillers are made from synthetic polypeptides that spontaneously form gels at low concentration in water. They're prepared by a robust and scalable polymerization process, similar to how commodity plastics are made, which is much less costly compared to sequential peptide synthesis. These hydrogels have many features that we feel make them excellent candidates as dermal fillers. Notably, their synthesis is scalable and economical. The properties are readily tuned, and they're designed with reversible crosslinks to allow disassembly and blood flow and injection through small gauge needles. And they've been prepared using natural amino acids to be degraded and resorbable and biocompatible in vivo. We expect that we can mimic the degradation times of existing fillers by tuning the characteristics of our materials. So how and why do these attributes allow our fillers to outperform current hyaluronic acids? In order to make their materials stiff enough to replicate the stiffness of human tissues, hyaluronic acid polymers have to be permanently cross-linked into individual gel particles over 300 micrometers in diameter. In contrast, our polypeptide materials can achieve the same level of stiffness with reversible cross-linking. These differences are readily verified by a simple experiment. Hyaluronic acid fillers taken directly off the shelf cannot pass through a 5 micrometer filter syringe the size of a capillary. The particles are just too big. However, our fillers with equivalent stiffness pass easily through even a 1 micrometer filter syringe and reform gels on the other side. Meaning, if our filler is ever inadvertently injected into a blood vessel, it should easily traverse the capillary and eventually be degraded or excreted from the body without causing harm or occlusion. 2, hyaluronic acid polymers have numerous negative charges that attract water, causing these materials to swell significantly for weeks after injection. In vitro, hyaluronic acid fillers have been observed to swell to 300%, similar to that 100% additional swelling we saw earlier with the animal experience in vivo. In contrast, our fillers are overall neutral, meaning they draw in less water and only swell about 25% when left in aqueous media for several days. This translates to a reduced risk of external vascular compression and more predictable augmentation in patients, likely leading to higher patient and provider satisfaction with the product. We've also injected our materials into wild-type rats and observed no clinical or histological inflammation, erythema, or irritation even out to 62 days. In other work, very similar materials have been injected into mouse central nervous system tissues and IV up to 2 grams per kilogram with minimal inflammation and no adverse reactions over time. We therefore expect that our products will not cause the occasional chronic or delayed irritation that's observed with hyaluronic acid fillers. So in summary, we believe that our fillers have the potential to outperform current fillers and address their 3 most notable limitations. So why is this even a market worth entering? Almost 3 million separate dermal filler procedures were performed in the United States last year by plastic surgeons. So this doesn't count the numerous filler procedures performed by other providers like OB-GYNs, cosmetic dentists, facial plastic ENTs, etc. As you can see from the chart, the majority of these procedures are hyaluronic acid filler materials. And the market is growing rapidly based on growing evolving body ideals, somewhat thanks to social media, as well as an increased desire for minimally invasive options. The market is currently a multi-billion-dollar a year market with an over 10% compound annual growth rate. We emphasize that this entire market is cosmetic and pay out of pocket, meaning that these products are immune to insurance coding issues and the price is set by the manufacturer without risk of being manipulated by insurance providers or Medicare. And again, this slide just demonstrates that this is an enormous multi-billion-dollar market, and 76% of the current market is occupied by hyaluronic acids, which we believe our material will outperform. The light blue on this slide is the hyaluronic acids. Comparing our filler to competitors, we note that only our filler offers all of these distinct advantages. It's safe, it's cheap to produce, predictable volume, tunable stiffness. Tunable stiffness is important because different parts of the face benefit from different stiffness and viscosity, and we can mimic the stiffness of the entire spectrum of fillers available on the market by modifying certain characteristics of our molecule in order to allow us to compete successfully in all areas of the face. Current fillers are dangerous if it's accidentally injected intravascularly. Other fillers also have the same vascular safety concerns and typically aren't as tunable as the hyaluronic acids, which is why they only occupy a fraction of the market. Dermal fillers are regulated as Class 3 medical devices. This is a major advantage for us because it means we will enjoy market exclusivity if we are the first to market for the currently vacant indication of periorbital injection or other currently off-label indication. Other products will not be able to reach the market through the less burdensome 510K Me Too pathway. We anticipate that we can submit a PMA for indications including all areas of the face in a single clinical study. Thank you. And at this time, we'd like to entertain any questions.
My name is Dr. Lee.
All right. So we're opening up the floor for the Q&A. If you have any questions, we'll take the next couple of moments for you to ask any questions of the team. If not, we can move on to the next set of presenters. All right, so we got the first question from the crowd, which is, what do you consider your top 3 hurdles to overcome?
Right. I mean, obviously this project is going to, there are going to be significant hurdles to get this project to market. As we said, it's a PMA device. And so anytime you need to do, the standard, it's a novel material. So there are going to be a host of GLP biocompatibility studies in addition to a clinical trial. So those are going to be very expensive. So that's going to be a significant hurdle. Another significant hurdle for this project, depending on our exit strategy, is the fact that, the dermal filler market is not, I mean, to some extent it's provider dictated, but, patients know the products, providers know the products, is brand loyalty. And Allergan, which makes Juvederm, occupies the majority of this market. And they have significant advantages because they also make breast implants or other products that these providers use and can kind of create situations where they have discount rates for providers who use a host of their products. And so entering the market will obviously be very difficult if we intend to make this product and push a company forward without being acquired. I would say those are kind of 2 of the main hurdles. Tim, would you think there's, and then we also are, relying on scaling up synthesis and finding manufacturers so we don't get limited to a single manufacturer. I don't know what you would say, or maybe.
Yeah, manufacturing, safety, and showing we do have superior properties. Those are probably, in a nutshell, 3 things.
All right, so that answer took a little bit more time than we had allotted. This team is going to be here for the rest of the conference. Please reach out if you'd like to network with them on Jujama and join us at the Innovation Fund information hour at lunch if you'd like to get any more details. But we're going to move on to the next team led by Dr. Lena Nathan with novel vaginal drug delivery.
My name is Dr. Lena Nathan, and I'm very proud and excited to present to you our project, Novel Vaginal Drug Delivery. We have a public health problem. Vulvovaginal atrophy affects more than 50% of postmenopausal women in this country. That number is about 32 million women. About 50% of these women will seek treatment for their vulvovaginal atrophy. And by 2030, 60 million women in the United States will be older than 50, leading to them suffering from vulvovaginal atrophy. We have a need for novel therapeutic methods to treat women with this condition. The current products on the market have poor patient compliance because there are flaws in the delivery systems. And surprisingly, there have been no significant improvements in vaginal drug delivery over the last 20 years. We have a solution, a user-friendly, biodegradable drug delivery system. So what is this problem? Vulvovaginal atrophy affects postmenopausal women. It is chronic and progressive. It is due to the lack of estrogen in the vaginal tissue. As you can see in our diagram here, a healthy vagina has thick epithelium, which is well lubricated. As women go through menopause and have less estrogen, their tissue becomes dry and thin in the vagina. This leads to dyspareunia, or painful intercourse, vaginal dryness, itching, and irritation, as well as urinary symptoms like dysuria or painful urination, recurrent urinary tract infections, and incontinence. The best treatment for this condition is vaginal estrogen. However, there is poor compliance with the current therapies. The therapies are mostly creams or pills, and they are messy and painful to use. They require frequent administration. They are expensive, and there is environmental waste with plastic applicators. Here is our team. Dr. Tamara Grisales is an assistant clinical professor at UCLA in the Division of Female Pelvic Medicine and Reconstructive Surgery. I am also an assistant clinical professor at UCLA in the Department of Obstetrics and Gynecology. Dr. Song Lee is the chair of UCLA Bioengineering and a chancellor's professor. He is the one who has the lab where we are creating this product. Dr. Mahdi Hassani works with him in his lab as a project scientist. We have already received funding for this project from the UCLA Innovation Challenge, our chair of Obstetrics and Gynecology, the Faculty Innovation Fellowship, and the UCLA Innovation Fund. So what is our innovation? RestoreFem. This is the next generation vaginal drug delivery method. It is an 8mm disc created using estradiol, as well as polycaprolactone, or PCL, an FDA-approved biopolymer. This disc, as you can see in the diagram, as a white dot, will sit at the top of the vagina to release medication. It is bioabsorbable, mess-free, and easy to insert. It's long-lasting and environmentally friendly because it does not require frequent use of plastic applicators. Our solution offers a clean solution for long-term and sustained medication release by using FDA-approved biodegradable polymers. There are two ways in which we can create our disc. One is the melt mixing method, and the second is the solution mixing method, and both of these methods are equally efficacious. A brief technology overview. We have studied in vitro the release of estradiol from the PCL disc, and we know that there is a constant and steady release of estradiol for up to 80 days, as demonstrated in these graphs. What is our competitive landscape? So there's the E-string, which is a silicone ring that sits inside the vagina for up to 3 months, releasing estradiol. This ring is not bioabsorbable and must be removed from the vagina and replaced at the end of the 3 months. There are several short-term bioabsorbable products, such as Estrace or Premarin cream, Vagifem, which is a pill, and Imvexi, which is a soft gel capsule. Our product will be the first product on the market which will provide long-term release and be bioabsorbable. Here is more information about the competitive market. So the E-string, as I mentioned, is a ring, and it actually lasts for 3 months, but is not bioabsorbable. The rest of the products on the market all require daily administration for 2 weeks, and then twice weekly administration after that. Ours will be the first product where you can use an insert every 10 to 12 weeks and not have to remove anything. As you can also see, there is insurance coverage for some products and not others. Our goal is to have our product covered by insurance. What is the market size? The total addressable market for vulvovaginal atrophy is more than $51 billion. The serviceable addressable market is $3.7 billion. This is how much women are spending on vulvovaginal estrogen therapy at this time. Our serviceable obtainable market, we believe, is at least 15%. This leads us to conclude that we can capture about $555 million of this market. The market growth is estimated at 7.7% CAGR, and our initial target market is women aged 45 to 65 who have private health insurance and are seeking treatment. We have been involved with customer discovery. We have completed the local levels of NSF I-Corp and are applying for the national level. We are also conducting a patient survey to understand how women feel about vaginal medications. We are asking about symptoms as well as satisfaction with the current products being prescribed. And we have also included fertility and vaginal infection medications. In terms of intellectual property, we have filed a utility patent for the vaginal drug delivery system. Our innovation is based on tunable flexibility, shape of the device, microstructure function, biocompatibility and bioabsorbability, as well as the time dose release capabilities. Our PCT patent application was filed in October of last year. How about our regulatory pathway? So the FDA has told us that this will fall under the 505(b)(2) regulatory pathway. This is an abbreviated approval pathway for drug products. Our pre-sub has been completed and the FDA has given us clear direction for the future. We remain in close communication with the FDA as we're planning our next steps for preclinical and clinical studies. Here is where we are with our development. We have completed milestone 1, which is to optimize drug delivery formulation, develop our clinical protocol, and correspond with the FDA. We're currently in milestone 2, which is to adjust our product design based on the FDA's feedback. Next, the FDA has recommended that we perform in vivo safety studies and biocompatibility studies in rabbits, and this will be our next step later on this year. After this is completed, we will pursue GMP manufacturing in our IND. And then finally, we will incorporate a startup, license back the technology, and raise Series A funding. There are other potential applications for this method of vaginal drug delivery. In IVF and infertility patients, all women require vaginal progesterone. Oftentimes, this is required up to 3 times a day. This is, as you can imagine, is very messy and inconvenient. We have created a PCL-based disc that can be used to deliver progesterone. And again, in vitro, it has been demonstrated to have a constant flow of progesterone from this disc in order to provide excellent therapeutics for these women. For vaginal infections, we have created a PLGA disc. This is another FDA-approved biopolymer. This can be used to deliver antibiotics, such as metronidazole for bacterial vaginosis or fluconazole for vaginal yeast infections. As you can see in this data, these work over a time course of about a week, which is perfect for medication delivery for these conditions. Thank you. We'll be happy to answer any questions.
All right. Thanks, Dr. Nathan and team. I'm going to open up the floor for questions for the next few minutes. There we are. Dr. Nathan's on. All right. So the first question from, hey, Madi. First question from the floor we have is, I'll just read it verbatim. If there is no plastic applicator, how do you ensure proper placement for optimal efficacy and prevent the device from being flushed out by increased vaginal secretions and decreasing size of the device over the extended period of time that it needs to stay in place? Did that question make sense?
Yes. So we do anticipate needing an applicator for this product, but we also expect that the applicator will only be used once every 2 to 3 months, which is much better than using it every day or twice weekly.
The next question that we have is about the bioabsorbable polymer. Is the bioabsorbable polymer a proprietary blend, or are you planning on licensing it from a supplier?
Polymer, maybe I can answer this question. This polymer is commonly used in being used for other products in clinical trials. I think we can get commercial available polymer, and we could use this polymer and basically make the product with different kind of procedures as described in the presentation.
All right, and here's one dear to my heart. What are you considering as exit strategies?
Tamara, do you want to answer that question?
Well, I think ultimately our goal here is our short-term goal, short to medium-term goal is to license back this technology and create a startup as in terms of kind of final exit strategy. I think that we have a little ways to go to see where we get with our testing and products. I don't know if you have anything to add to that, Lena.
I agree. I think eventually, we do want to create a startup and take this as far as we can on our own, but eventually I believe we will get acquired by a larger pharmaceutical who has the resources to bring it all the way to market.
That wraps on the questions. So thank you so much, Novel Vaginal Drug Delivery team. Just as with the last team, they're all available on the Jujama platform. So if you feel inclined to reach out to them, please do. And if you're interested in further information, please join us at the lunch break at the Innovation Fund kiosk. From here, we're going to move on to the next and final presentation, Dr. Ron Harper with vibratory sensory stimulation, a novel therapy for sleep apnea.
We developed a novel means to manage sleep apnea, a very common and serious medical condition. We use mechanical vibration to activate cranial nerves. We are a group of neuroscientists and pulmonologists who have spent most of our lives devoted to examining the brain processes underlying disrupted breathing during sleep. The problem with sleep apnea is that the current interventions are very poorly tolerated. They're unsuitable for certain forms of apnea, and they fail to address the principal concomitant of sleep apnea, which is hypertension. We've developed a device which is safe. It uses simple vibration. It's non-invasive. There is no electricity going to the skin. It's very well tolerated. In fact, it induces sleep. It addresses all types of sleep apnea, and it also helps to regulate blood pressure. There are two forms of sleep apnea. Obstructive sleep apnea, in which breathing in the diaphragm continues, but the upper airway muscles collapse, and that leads to cessation of airflow. The second type is periodic breathing, or central apnea. In this case, the brain triggers the respiratory muscles to breathe, but then stops that drive, and then triggers the breathing motions again, and then stops it, and then again breathing, and that continues for very long periods of time. The problem we see down below, and that is that oxygen saturation drops precipitously. And you can't have that. You can't have these kinds of drops in oxygen saturation without encountering brain injury. Part of the problem is the prevalence. Nearly 40 million Americans have sleep apnea. Not only that, 2/3 of heart failure patients have central sleep apnea, that is periodic breathing. The whole world suffers from this problem, and the principal intervention, continuous positive airway pressure, that market is growing at an annual rate of 6%. The current interventions are really not adequate. The gold standard, continuous positive airway pressure, CPAP, is tolerated only by 1/3 of OSA patients. It's uncomfortable, as you see here in the screen. It's large. It interferes with bed partner intimacy. And worst of all, it doesn't work for periodic breathing. And it doesn't work for long-term blood pressure control. There are other techniques. You can move the mandible forward using an oral appliance. That's only good for mild to moderate sleep apnea. And it's also useless for periodic breathing. And it also introduces the potential for temporomandibular joint pain. We have another intervention. We electrically stimulate the nerves that drive the tongue and upper airway muscles. In about 1/4 of the patients, that doesn't work. It involves invasive, costly surgery, and there's a huge infection risk. And of course, it's expensive. What we do is use an approach of recruiting cranial nerves that will affect breathing. And we went for a spot on the body where most of the cranial nerves were located, and that is the ear and ear canal. We vibrate those structures, and the brain sends signals down to the diaphragm and to the upper airway, including the tongue, to open the airway, preventing obstructive sleep apnea. And of course, since both diaphragm and upper airway muscles are stimulated, we can get at the issue of periodic breathing. We use a device made of silicon with a attached magnet and attached metallic rod to stimulate the various cranial nerves that are in the ear canal. The vagus, number 10, trigeminal nerves, cranial nerve 7, cranial nerve 9, which is the nerve for blood pressure control. In addition, we also vibrate the cervical nerves 2 and 3, which form part of the phrenic nerve to the diaphragm. The device is powered by a vibratory motor, which you see here, and through this power supply consisting of 2 AA batteries. We use various forms of device here in a pediatric patient with periodic breathing and oxygen desaturating to the low 70s in these stop breathing periods. The stimulation, saturation returns to the high 90s, and as you can see, the breathing is continuous with no periodic breaks. This is an adult, 33-year-old male, congenital central hypoventilation patient, a disease in which all breathing stops. He has both CPAP and a phrenic pacemaker to drive breathing, but those, as you see in the left-hand side, don't adequately support oxygen saturation. But with the vibratory device, you can see the extent of breathing movements as well as the high oxygen saturation that's maintained. This individual also, when he enters REM, loses blood pressure, which of course affects perfusion and can cause brain injury. Here he enters REM and drops blood pressure, but you can see that the vibration device maintains that both that systolic pressure and diastolic pressure. We have developed a number of devices over the years. 2 patents have been granted, 2 are pending, and a European pending grant, which should be signed next week. The FDA has classified one of the devices as class 1, low risk, and the UCLA Clinical Engineering and UCLA IRB have labeled it no significant risk. Because we address a number of cranial nerves, we can use the device for several other medical indications. And we've used the device for migraine in over 60 patients with huge success. We've reduced seizures by about half in a very high-risk population. And we're working with University College London to study breathing patterns in COVID-19. They lack breathing drives to CO2 and O2, and we feel we can provide that drive with this device. We've completed a neonatal study, published it, a migraine study, we have IRB approval for a new study on adults, and we have costed the funds needed for OSA and for heart failure components of the study. The practical intervention is backed by a number of theoretical studies in the past and rests on solid physiological principles. Thank you.
All right, I see Dina on, and so that usually means that it's time for us to move forward. Is that right?
It is. I'm so sorry to cut this off. We're about 5 minutes over.
Okay. Well, thank you so much, Dr. Harper, and thank you for joining us today. Same as with last time, It looks like we have a number of questions, so I'll just make sure to note all of those and the folks who have asked them. And please meet us at the Partnering Center for the Innovation Fund. Thank you.
All right, thank you.